you should draw the DNA molecule in the very limited time that you have during the final exam when all you see is a blank sheet of paper in front of you. And of course it's going to take me a little longer than you because I got to fiddle with these pens and I don't have colored pencils, but we're going to try. So let's begin. Are you trying to get your sound on the video? That's great. It's like so festive and holiday. It's like the sleigh bells. I like that. Okay. So anyway, for Nolan and others, see I got your name in there. I don't have a pencil, so what you're going to do is just use a regular pencil. And I think the best thing to do when starting a DNA molecule is to draw little faint guidelines that show that we have this double helix structure. And I'm going to erase these guidelines as we go along or at the end of the process, but that's going to keep me oriented. So that fills up the page, makes my full page. Here's my page of paper. If I were you, taking the final exam and having to draw a DNA molecule from memory, it would look something like that to start with. And again, this is my pencil, and I don't have a pencil. You know what I'm talking about. Okay, so let's begin. And I'm going to begin with the color purple. And I'm going to put a series of dots right across from each other. A dot here and a dot there. A dot here, a dot there. A dot here, a dot there. A dot there, a dot here. And etc. all the way down the page. And I'm going to label that as being the phosphate molecule. Okay? And in fact, you might want to put a little P next to each one of those dots, just so everybody knows that you're talking about a phosphate molecule. And if you notice, they're right across from each other. I think I said that already, going down the background. Okay, so that takes care of, on your bucket list, that takes care of two things. Check, we got the double helix shape started. Check, we got the phosphate molecules. Aren't those on the checklist? I think they are. So let's move on to the next one. And for the next one, we are going to draw, for you baseball fans, we're going to draw a little picture of home plate. Okay, there you are, home plate. Let's put another one over here. Let's put another one over here. And we got another one right over here. And finally, we got another home plate right down there. And if you notice, I've just followed one of the strands down the line here. And I also have home plate in these pointed upward. Now, of course, what is home plate? Home plate, we can put an S in there. This is the sugar molecule that helps make up the backbone. And we know the name of that is deoxyribose. And of course, deoxy means there's one less oxygen than ribose. And all carbohydrates, sugars and starches, end in the ending -ose. So we know automatically what a sugar is, or a carbohydrate is, by its name. So in parentheses, I'll put sugar, the sugar, and there they are. Now, going down the other side, something very weird happens. We talked earlier in our review about the anti-parallel orientation of the DNA molecule. So one strand actually goes up, and the other strand actually, I'm not doing that right, am I? Good thing I have a trusty eraser. That's a pitiful sugar molecule there, Mr. Perry. Okay. So let's actually make it look like it's pointing downward this time instead of being in such a big hurry. And there it goes, pointing downward. And there's another one over here, pointing downward. And that's just barely room for one more down there, pointing downward. And those are the sugars on the other side, the other strand. So we have one strand running up, one strand running downward, and that's the anti-parallel orientation of the DNA molecule. Okay, so we got that, we got the sugar, we got the phosphate, and together, we know what that is. What is it when you have all those sugars and phosphates? They give that kind of nickname. It's called the backbone, exactly right. The backbone. Just like you have a vertebral column going down your back. Okay? So there's another thing I can check off, a couple more things to check off the bucket list. And now, for you colorful characters, I'm going to choose the color orange to start with, okay? And in orange, I'm going to put right here, connected to that sugar molecule, I'm going to put a T and a couple of dots. And maybe just to have fun, I'll put a T down here, connected to that sugar, and I'll put a couple of dots. All right? Okay. So what we got here, when you take that T, that S and that P, we got the sugar, the phosphate, and the base, the nitrogenous base. We have thymine, and since we have all three parts, it's called thymine nucleotide. Okay, a nucleotide is made of three molecules, the sugar, the phosphate, and the base. The base is thymine, so they call it thymine nucleotide. All right? So we have another one down there, just to kind of fill it out a little bit. Now, it turns out that according to Erwin Shargaff, who added all this stuff and weighed them out, he found that the amount of adenine was pretty much darn close to being equal to the amount of another base, which I'm doing in brown, and that's called adenine. And I'll put an adenine there to match up with the thymine, and adenine there to match up with the thymine. So that's adenine. And if you take the phosphate and the sugar and the adenine base, you have adenine nucleotide. Okay, so we've knocked off 2 of the 4 nucleotides off of our checklist. And let's go down the line now, and I'm going to choose the ever-festive holiday color green. Okay? And let's put a big G here. Put a big G. And that G is going to stand for guanine. And if we count the phosphate and the sugar, it would be guanine phosphate. Guanine nucleotide. Okay? Well, we put some more. Let's put another one over there, just because it's blank. And I'll put another one over there, just because it's blank. We have to fill in something. Okay? All right. Now, we know that adenine and thymine bonded together, so guanine is going to bond with what? Cytosine, exactly. So G's and C's go together, A's and T's go together, and that is cytosine. And if you count the phosphate, the sugar, and everything, it's cytosine nucleotide. So everywhere I have a G, I got to put a C, and everywhere I have a G, I got to have a C. A C's got to have a G, a G's got to have a G, A's got to have a G. Now, as you go down the backbone, as you go from top to bottom, your genetic code, the thing that makes you what you are, is this sequence of code letters. So your code to make you what you are might be T, C, G, A, G in this molecule. That is the little section of your genetic code. You read it right down the page, T, C, G, A, G. Now, it could be that there's some instruction, some enzyme that says, oh, no, we're going to read the other side, in which case your genetic code would be A, G, C, T, C. So you just read the letters on one side or the other, and there's an enzyme that tells it what to do. Okay, on our bucket list, the only thing that I think we have left to explain, and while we have time, is these little dots in the center here. In between the nucleotides are some special kind of bonds, and those are the weak hydrogen bonds. Okay? So the little dots in between the molecules, you see them in between the base pairs. The T's and A's are weak, the G's and C's are weak. What does weak mean compared to what? Compared to strong ones. They're the ones that are most likely to break. That's why they're called weak. So all the rest of the bonds, all the rest of them that I drew in black ink over here, those are actually strong bonds or covalent bonds. So I think that pretty well wraps up the structure of DNA as you would draw it on a full sheet of paper without any notes from memory with your colored pencils on test day. Thanks very much.